Bacterial shape impacts both nutrient uptake, i.e., how they take up nutrients, and also motility, how they move. And even though we're only demonstrating 4 categories here, there are many more, including organisms that are square shaped, hexagon shaped, and even star shaped. The first category that we're going to look at are the cocci, or the coccus. So examples of these include Streptococcus pyogenes, and Streptococcus pyogenes causes strep throat, and Staphylococcus aureus, which is a common cause of skin boils and abscesses. Note genus capitalized, species lowercase, all in italics. Now these are round in shape and they can form both clusters and chains. So strepto actually means that these organisms form into chains. So streptococcus means chains of circular organisms. On the other hand, staphylo means they form into clusters. So staphylococcus is clusters of round-shaped bacteria. The next shape that we're going to look at are the rods. Now, these used to be called bacilli, so you might come across that terminology. But bacilli is also a genus name, so we've gotten away from that, so now that's why we call them rod shapes. Example here that we are looking at is Clostridium tetani, and Clostridium tetani causes tetanus. Our third category are the Spiralia, and these are large spiral-shaped rigid cells usually found in organically rich stagnant freshwater. But so far, we have found no human diseases that we're aware of. A great example here is Spiralium volutans. And we quite often study these because they have some really great flagella, which are used for motility. And we'll come back to flagella a little bit later this semester. The fourth category is the spirochetes. These are less flexible than the Spiralia that we just talked about. They're very tightly wound up, like they're little corkscrews is the really easiest way to classify them. Example here is Borrelia burgdorferi, and Borrelia burgdorferi causes Lyme disease. To see these shapes and other morphologies, we need to be able to enlarge an image. This is accomplished through a series of lenses. These are referred to as compound lenses. So you'll see the term compound microscope, for instance. And there are 2 sets of lenses. The first set of lenses are referred to as the oculars, and those can increase the image by 10 times, which we see here. This is the most common, 20 times or even 25 times. So that's 25 times what the naked eye would see. This can be further enlarged by the objective lenses, of which there is quite frequently a combination of different sizes. So we've got 10 times here. We'll often see a 4 or a 5 times, a 40 times, and as we see over here, 100 times. Magnification is determined by taking the ocular and then multiplying it by the objective. So you determine it by multiplying both lenses. For example, if we take 10 times for the ocular and multiply it by 10 times for the objective, we conclude with 100 times magnification. This is not the magnification. This is the magnification here. Over here, we have 10 times 100. So this is 1,000 times magnification. So here at 100 times magnification, we can see something here underneath the microscope, but not really the detail that we might need. Here at 1,000 times magnification, however, we can see the individual red blood cells and the individual white blood cells.